to start. It doesn't really matter if
We're good. We're starting.
Okay. All right. So this overall reaction should look very familiar to you. It should look a lot like the reverse of the photosynthesis reaction. Remember that glucose has a chemical formula of C6H12O6. So we get C6H12O6 plus 6O2 yields 6 CO2 plus 6 water plus energy in the form of ATP. And we get approximately 38 ATP molecules. That is a maximum ATP. We almost never actually get 38 ATP. Now, if you're looking at this overall reaction, it is not a single chemical reaction. It's actually a summary equation for many, many smaller reactions. Now, this is the summary equation of one type of respiration, specifically aerobic respiration, and you can tell because it has oxygen in the reactants. We're going to be talking about two different types of respiration today, anaerobic and aerobic, and this is the aerobic version. This is the more common version of respiration. If you were to just generally describe what is respiration in the simplest terms to, fifth grader or a little cousin or something, you would say it's the process where cells extract energy from food. So in photosynthesis, in the unit we just finished, we had food being made by photosynthetic organisms like plants and algae, and now we're breaking that back down. Okay? So the very first step of cellular respiration, regardless of whether you have aerobic or anaerobic respiration, is always glycolysis. Glycolysis is the breakdown of glucose. You always know anything with glyco is referring to carbohydrates, usually glucose. Remember you learned glycoproteins, glycolipids, right? And if you see lysis, it means breakdown or destruction. So we're breaking down glucose. This process is the most ancient of all the processes that we're going to be talking about today in terms of evolution. When you break down glucose, the product is two pyruvates. Another name for pyruvate is pyruvic acid. Those two can be used interchangeably. So I will go back and forth using one or the other. Two products are ATP, you know what that's for, it's to do work in the cell, and two NADH molecules. Just like when we made NADPH in the light reactions, and I told you we were going to use it later, for now you're going to have to just trust me that this molecule, NADH, is going to be used later in the electron transport system, which we will cover in a couple weeks. But this is going to be very important at a later date. Be really careful because lots of students on quizzes and tests get NADH confused with NADPH, and we tend to put both in word banks. So just be very, very aware of that. Think P for photosynthesis, even though that's not what the P in NADPH stands for. It's just a little memory trick. So first I want to talk about this side of the diagram. And by the way, the only thing in this diagram that actually represents an actual structure in the cell is this blue structure down here, which is a mitochondrion. And we'll get back to that later. But this is just a flow chart that just shows you an organization of information. And so we have our anaerobic side and our aerobic side. It's kind of like, a choose your own adventure story with 3 different possible outcomes, depending upon the organism you have. So anaerobic respiration is respiration when no oxygen is present. And there are actually some species of bacteria, we actually call them strains of bacteria, that are obligate anaerobes, which means they only grow and reproduce when there's no oxygen. All of the obligate anaerobes are all bacteria, but it's fairly rare to be an obligate anaerobe. The 2 examples that I can think of that you may have heard of that relate to obligate anaerobes are, if you've ever heard of gangrene, which is can happen if you get a war wound, if there's a very deep penetrating wound and there's flesh that dies. And if the flesh is dead, no oxygen is supplied to that area. And so if this particular type of bacteria gets deep into that wound, it will fester and you will get gangrene and you can lose a limb, or potentially if the person doesn't lose the limb, they can end up dying. So that's why sometimes in old time war movies they'll have to have a limb removed. Of an obligate anaerobe that you may have or be familiar with is if you've ever heard of the type of food poisoning called botulism. It actually happens in canned food when they don't properly heat the food, and then they remove the air from the can, and then you have an absence of air, and then you have the right bacteria, and they're actually in the perfect environment to thrive. They've got food and no oxygen, and so they reproduce in great numbers and produce a lot of toxin, which then if you eat it can be very, very deadly. Antibiotics don't work because you don't actually have an infection. You've actually just ingested a whole bunch of poison. So if you ever see a can that has a funny shape, particularly if it's bulging outwards, it's probably because you have an obligate anaerobe in there, and it's fermenting and it's producing carbon dioxide, and when the carbon dioxide builds up, it expands and changes the shape of the can. The same thing is true, not necessarily botulism, but if you've ever eaten one of those little fruit cups and you've seen that the little plastic thing that you peel off the top is kind of shaped like a dome, and you like open it and it makes a really loud noise, that's probably because fermentation is going on. Now, with alcohol fermentation, this is one of the types of anaerobic respirations, and it happens in yeast or bacteria. And by the way, yeast are not obligate anaerobes, but we'll get back to that in a second. Some bacteria are and some are not. But alcohol is produced and carbon dioxide in some cases, and the other alternative one is lactic acid, but we'll get back to that in a second. So alcohol, well, ethanol has 2 carbons and an OH group. There are other types of alcohol, but if somebody just says alcohol, they're probably talking about ethanol. This is the kind of alcohol that's in alcoholic beverages like beer and wine. And with this type of fermentation, each pyruvate is broken down into an ethanol and a carbon dioxide. So 1, 2, 3 in terms of keeping track of our carbons. The carbon dioxide is actually what makes bread rise, and if you've ever looked at a slice of bread really closely, you actually see bubbles in it. Once you bake the bread, the alcohol evaporates. So there's no actual alcohol left in the bread once you have put it in the oven. Ethanol is so toxic that you can only get about 9 to 12% alcohol, or I think, well, 12 is the absolute maximum in any wine or beer before the bacteria or yeast themselves die from their own toxic waste. The other types of alcohol that they sell that actually have higher alcohol content, they actually have to remove the water by separating them based on their differences of boiling point between water and alcohol. You can actually evaporate the alcohol and then recondense it separately so that it can be condensed. But we are the only species that voluntarily poisons ourselves. This is very highly toxic. Okay. Now, the other alternative type of anaerobic respiration is called lactic acid fermentation. And by the way, somebody after reading the bio preview yesterday asked me, is anaerobic respiration the same thing as fermentation? And yes, both types of fermentation are considered anaerobic because they don't use oxygen. They are sometimes used interchangeably, although the fermentation, if we said alcohol fermentation or lactic acid fermentation, that's being more specific because both of these would be considered anaerobic respiration. So with lactic acid fermentation, we have a 3-carbon molecule, and in a few minutes you'll actually see the exact structure, but we do not get any carbon dioxide with lactic acid fermentation. Now, for lactic acid fermentation, this happens in different species of bacteria. Again, we call them actually strains, but I didn't think you guys would know the term strains versus species. You'd be more familiar with the word species. But so far as I know, there aren't species of bacteria that can do both of these. It's an either-or proposition. Yeast always do alcohol fermentation. Some bacteria do alcohol fermentation, and some do lactic acid fermentation. Our tired muscles, if we're talking about our skeletal muscles, the voluntary muscles, when we run out of oxygen, I mean not completely run out of oxygen, but when we are low on oxygen compared to our oxygen needs, our muscle cells will switch over to lactic acid fermentation for short periods of time so that the oxygen we do have can go to our brain and other vital organs. Lactic acid can actually be reconverted back into glucose once you have enough oxygen and broken down by aerobic respiration. If you've ever been exercising and felt the burning in your muscles while you're exercising, that's that tells you that lactic acid fermentation is going on, although it's not that actual acid that's burning, making the burning feeling in your muscles. That does not count for the burning that you feel the next day, the soreness. That's something different. That's delayed onset muscle soreness, not necessarily related to the lactic acid fermentation. This tends to happen when you're running sprints, or if you're lifting heavy weights, or lifting lower weights at very high reps. Like if you've ever done like 100, 200 squats and started to feel your legs burn, you know you're switching into anaerobic respiration. All right, so we talked about obligate anaerobes, and we're about to talk about organisms that can do both. But before that, I wanted to bring up the fact that we only got 2 ATP molecules. And the 2 ATP molecules that we get from anaerobic respiration all occur in this first glycolysis phase. So you should be wondering, if you only get ATP in the very beginning, why bother to break pyruvate down any further, especially into something that's so toxic? And the reason is that once you produce NADH, you have to use it up and remove the hydrogen to regenerate NAD+, so that glycolysis can continue. So the whole purpose of breaking pyruvate down into ethanol and carbon dioxide, or breaking it down into lactic acid, is to use up the hydrogen, pull that off, so that you get NAD+ back, so that more glycolysis can occur. So without these other chemical reactions, this very first important chemical reaction would come to a grinding halt. Okay, now I want to talk about some organisms that can actually do either anaerobic respiration or aerobic respiration. Yeast are an example, and many species of bacteria are also able to do either aerobic or anaerobic respiration. They are referred to as facultative anaerobes. They can do either one. Now be really careful not to call them facilitated anaerobes, just because we recently learned about facilitated diffusion. Also, I always have students ask me, well, why didn't they call them facultative aerobes? Or is there such a thing as a facultative aerobe? There is not such a thing as a facultative aerobe that I've ever heard of, and I don't know why they decided to call them facultative anaerobes, since they could switch back to either one. I don't know why, but this is what they're called. Make sure you know this term. We do a whole lab on facultative anaerobes, but for some reason, by the time we get to the test, everyone forgets what a facultative anaerobe is. So I would highlight that. Now I want to talk about obligate aerobes, like us, and actually any multicellular organism is an obligate aerobe. It won't surprise you, because any multicellular organism is going to require more energy than a single-celled organism. Now the ones that can switch back and forth, the single-celled organisms that can do either, if they have no oxygen, they'll produce enough energy to survive, but if they have oxygen, they'll have extra energy and they will use it to reproduce. So the population will grow exponentially when there's oxygen around, because that's what the extra energy can be used for. It may surprise you that plants are obligate aerobes. It shouldn't surprise you when you think about the fact that plants produce a lot of oxygen. There would be oxygen around, and they would use the more efficient version if they could, and they do. Now, it's a common misconception that animals respire and plants photosynthesize, and while that's not really wrong, it's not a complete picture. Plants respire and photosynthesize. It's also a common misconception. Some students are like, okay, I get that plants respire, but plants only respire at night when they're not photosynthesizing. And that's not true either. All organisms respire all the time unless they're dead. So plants photosynthesize during the daytime, and they respire during the day and the night. So the second and third stages of cellular respiration, which I'm just going to mention today and tell you their immediate products, occur in the mitochondria. If you see mitochondrion, that's singular. If you see mitochondria, it's plural. So there are two more stages. The first one is the Krebs cycle, named after Johann Krebs, who actually figured out all the stages. And the Krebs cycle produces 2 ATP and 6 carbon dioxide. The third phase is referred to as ETS, And ETS stands for electron transport system, or sometimes it's referred to as ETC, the electron transport chain. Either one is acceptable. I sometimes say electron transport system. I sometimes say, well, I usually say ETC or ETS, but I kind of go back and forth between using those terms. The ETS is where the oxygen is finally used. Water is going to be produced. This should remind you sort of as the reverse of the light reactions. And it's actually very, very similar to the light reactions. And when we show it to you in a couple weeks, you're going to be very familiar with it. There's going to be hydrogen ions moving across a membrane through ATP synthase. And instead of water being split to make oxygen, you're going to actually have oxygen picking up hydrogen to make water. But 34 ATP are made during the ETS. So there's a net gain of up to 38 ATP, and students can never figure out where all those 38 come from. But it's 34 from ETS plus 2 from the Krebs cycle. And they always forget that you still have those 2 from glycolysis, because glycolysis is the beginning stage of both aerobic and anaerobic respiration. So why do we get so much more ATP when we respire aerobically? Because the glucose is broken down all the way. In anaerobic respiration, you have a partial breakdown because you still have carbon chains. Here's 2 carbons in a chain, 3 carbons in the chain in the case of lactic acid. So if you still have carbon chains, even though they're smaller carbon chains than what you started with, there's still some untapped potential energy there. And in fact, you can actually set alcohol on fire. I don't recommend it, it's not safe, don't try this at home. But it can also be used as a fuel. There are cars that run on ethanol. So now I'm going to go into detail in this section. I'm not going to go into the details of these 2 sections today. If you walk out feeling like, oh, I don't know much about the Krebs cycle and ETS, that's because I haven't explained it to you yet, and we will go into more detail on both of these, not next week, but the week after next. But now, anything on this front page is fair game for the quiz you have coming up next Friday. Okay, now aerobic respiration is more recent in evolution than anaerobic respiration, and it's also more recent in evolution than photosynthesis, which should make sense because photosynthesis is what produced oxygen in our atmosphere in a high enough level for aerobic respiration to occur. So we would have had anaerobic respiration first, then photosynthesis, and then aerobic respiration in terms of evolution. All right, so now it would be very helpful to have out a highlighter, because I'm going to tell you which parts of this you definitely need to know and which parts of it you need to be able to recognize. Okay? So I am obviously going to ask you at some point what glycolysis is. So I would highlight the word glycolysis, and I would also know where glycolysis occurs, because you know we always ask you where every process occurs in the cell. Where are we? So glycolysis occurs in the cytoplasm. You can also say cytosol. Every step of the reaction series that I'm about to show you requires enzymes, a different enzyme at every stage. If you take an upper level college biochemistry course, they'll make you learn the names of those enzymes. We're just making you know that enzymes are involved. A different one at every stage. Every chemical reaction has its own specific enzyme involved. We start off with glucose. I would highlight glucose. Now glucose is converted into a molecule of glucose 6-phosphate. If we need to add a phosphate, there's only one molecule we know that's able to do that, and that would be our old friend ATP. So ATP adds a phosphate onto the 6th carbon in the chain. That's why it's called glucose 6-phosphate. This 6 tells us exactly which carbon in the chain the phosphate has been attached to. So if ATP is used, we get ADP back because the 3rd phosphate is now on the molecule. It has lost one of its phosphates. And it's gone from adenosine triphosphate to adenosine diphosphate. A second enzyme converts glucose 6-phosphate into fructose 6-phosphate. Those molecules have the same molecular formula, but a different structural formula. And we told you a vocabulary Be really, really careful not to confuse isomer with isotope. Both will be in any word bank or answer choices on a test or quiz. An isomer is a molecule which has the same number and types of atoms in it, same molecular formula, C6H12O6, but a different structure. An isotope is an atom of the same, or two isotopes are an atom of the same element that have a different mass because they have a different number of neutrons. Not the same thing. So be really careful to make sure that, if we're asking you about isomers, that you don't say isotope by accident. Okay, so fructose 6-phosphate is converted into fructose 1,6-diphosphate by an enzyme and ATP. So the reason it is fructose 1,6-diphosphate is that there are two phosphates and they're on the first and the sixth carbon in the chain. Do not be intimidated by the long words. It's just a fructose with two phosphates on opposite ends. So at this point, you might be wondering how we've used up 2 ATP, and in the previous page I said that by the end of glycolysis, we've gained 2 ATP. We're not done yet. So it turns out that at the beginning of this process, you actually have to spend ATP to get the process started. By the end, we'll end up with some extra ATP, but we're not there yet. So at this point, fructose 1,6-diphosphate is broken in half by an enzyme into 2 molecules of 3-phosphoglyceraldehyde, also known as PGAL. We have 2 of them, and there are 3 PGAL. This number 2 tells you how many molecules we have, and this 3 tells you where the phosphate group is, that it's on the 3rd carbon in the chain. Now at this point, we have more phosphate coming in, and for the first time, it's not coming from an ATP. It's just inorganic phosphate, which is just PO4 dissolved in the cytoplasm of the cell. Also, what's happening is a molecule of NAD+, an ion, is pulling off hydrogen. We are not showing you the hydrogen in this diagram. You're just going to have to take my word for it that hydrogen comes off and NADH is formed. I would highlight NAD+, NADH in both of these places. I would highlight ATP and ADP. Words like fructose 1,6-diphosphate are not likely to be words that we ask, but they're likely to end up in the paragraph part that we write, not the part that we ask you for. So now we have 2 molecules of 1,3-diphosphoglycerate. This number tells you how many we have. The 1 and the 3 tell you that the phosphate is on the 1st and the 3rd carbon. They're converted into 2 molecules of 3-PGA. If you look at this molecule, there are 2 phosphates here, and now there's only 1. So at this point, and you'll also see that there's a squiggly line there that tells you it's a high energy bond that's attached to that phosphate. At this point, an ADP can pick up that last phosphate that it needs and become an ATP. So by this time in the reaction, we've actually broken even in terms of ATP. We used 2, we've made 2, but we're not done yet. Now, in order to understand what's going on in this diagram, I want to draw to your attention that we're going to follow the fate of 1 molecule, but there are actually 2 of them. We just didn't have room on the diagram for both of them. So this down here says follow 1 molecule on to the next line. So we're coming down to here. Now this says down here, remember there's really 2 of these, but we're only showing you 1 because there's not a whole lot of room down here. So we now have 2 molecules of 2-PGA, and it's converted into 2 molecules of phosphenylpyruvic acid by an enzyme, and then another enzyme converts phosphenylpyruvic acid into 2 pyruvic acid molecules. And if you notice, phosphenylpyruvic acid has phosphate, hence the phosphenol, and pyruvic acid does not. So at that point, each of these 2 molecules loses a phosphate, and you can make a total of 2 ATP. Students always say, well, how do you get 2 ATP? I only see 1 phosphate here. That's because you have an ATP made for each of these molecules, and remember, we have 2 of them. Now, we have 2 molecules of pyruvic acid. Here is the structure. I would highlight pyruvic acid. I would also highlight pyruvate. Those are the same thing, 2 names for the same thing. Now, in this part of the diagram, this is just a repeat of what was on the previous page. So, and the green top part, we're talking about anaerobic respiration. This says without O2. Up here, it says it occurs in the cytosol. If we have yeast or bacteria, the pyruvate is further broken down into ethanol and carbon dioxide. Remember, this is what makes the bread rise. This gets evaporated when you bake it. This is the kind of alcohol that's in beer or wine. Okay, stay away from it. If you have other strains of bacteria or tired skeletal muscle in any animal, instead of the pyruvic acid being broken down into alcohol and carbon dioxide, it is converted into lactic acid. With oxygen, depending upon if you have a species that can use oxygen, we have the second and third phase of aerobic respiration, which occur in the mitochondria, and they are the Krebs cycle and the ETS, and then we get 6 total carbon dioxide, 6 water, and 36 more ATP to add to the 2 we got during glycolysis. Okay, anything that you highlighted on here, anything in this area is fair game for a quiz, right? Because it's the same stuff that's on the front side of your note page. Now this too just is, it says here that there's 2 ethanol, but you'd also get 2 CO2. All right, this summary of glycolysis. This summary of glycolysis is fair game for the quiz. Anything on here you need to know. So what do we start off with? We start off with our friend glucose, C6H12O6. We use 2 ATP and get 2 ADP. We use 2 NAD plus and get 2 NADH. 4 ADP are converted into 4 ATP, and at the end, we get 2 3-carbon molecules of pyruvate, also known as pyruvic acid, and we get a net gain of 2 ATP, because if you make 4 but had to spend 2, you get 2 extra ATP by the end of the process.